In today's lecture, we are going to study about biological nitrogen fixation. As we all know, nitrogen is one of the limiting nutrient in the soil for agricultural production. That is why we have to depend on more nitrogen fertilizers like urea, which in long run make the soil unfertile. Biological nitrogen fixation can fix atmospheric nitrogen into the plant by means of an enzyme called as nitrogenase. Now it is mainly applied in agriculture to replace the chemical fertilizer. So in this lecture we can elaborate the biological nitrogen fixation that is happening in the plant system. What is biological nitrogen fixation? Diazotrophic microbes, biochemistry of nitrogen fixation and nitrogen fixation in root nodules are the topics that we are going to discuss today. What is biological nitrogen fixation? The process of conversion of atmospheric nitrogen to ammonia by microorganisms through a reductive process is called as biological nitrogen fixation. It accounts for about 70% of the total nitrogen fixed in the biosphere. It has been estimated that 100 to 175 million tons of nitrogen are fixed annually by the process biological nitrogen fixation. That is why we can save a huge quantity of chemical fertilizer as well as the cost of production for the farmers. The nitrogen fixing microorganisms are called as diazotrophs. This process occurs in the presence of an enzyme called as nitrogenase. The diazotrophic microbes can be categorized into several groups like free living aerobic bacteria, free living anaerobic bacteria, associative symbiotic bacteria, symbionts, endophytic and so on. Some of the examples for the free living aerobic bacteria are Acetobacter, Beringia, Klebsiella and Cyanobacteria. Free living anaerobic bacteria are Clostridium, D-sulfur vibrio species, purple sulfur bacteria, purple non-sulfur bacteria and green sulfur bacteria. Azospirillum species is a typical example for associative symbiotic bacteria, and symbiotic nitrogen fixation comprises of the well-known Rhizobium species, which has an unique association with legume crop, as well as Frangia, which is having association with non-legume tree crops like Casuarina. Endophytes are microbes that resides inside the plant's tissues. For example, Gluconacetobacter lives inside sugarcane, as well as Herbaspirillum from rice, sorghum and maize, as well as Azoarchus from rice. Symbiotic nitrogen fixers and their association with the host plants are very much beneficial for increasing agricultural productivity, especially this Rhizobium legume association, Bradyrhizobium soybean association, Azorhizobium sesbania rostrata association, Anabena sole with Azolla and Frangia with Casuarina. So in this picture we can see the stem nodules which is nodulated by the stem nodulating bacteria Azorhizobium colinodans. Let's now see the biochemistry of nitrogen fixation. The process of nitrogen fixation is mediated by an enzyme called as nitrogenase, which mediates the reduction of nitrogen to ammonia. Ammonia is the preferred nitrogen source for the plant system. Plant can take nitrogen either in the form of ammonia or in the form of nitrate. In the year 1960, nitrogenase enzyme was extracted from the anaerobic nitrogen fixing microbe Clostridium pastörinum. Later, it was isolated from most of the other nitrogen fixing bacteria as also. In this picture, you can see the structure of nitrogenase enzyme with all the subunits and different clusters. So, the nitrogenase enzyme is composed of two subunits. One is the iron protein unit, another one is the iron molybdenum protein. The iron molybdenum protein in turn have 4 subunits which contain iron as well as sulfur. This is otherwise called as dinitrogenase. So, in the picture we can very well know that while reducing nitrogen to ammonia, it requires 8 protons as well as 8 electrons and 16 ATP molecules are required. So, for one molecule it requires 8 and 2 molecules of nitrogen it requires 16 ATP molecules. So, the optimal ratio is 1 to 2 iron protein is required for 1 iron molybdenum protein for the optimal activity of nitrogenase. The reduction of nitrogen requires 8 protons and 8 electrons, totally 16 ATP molecules are required to reduce a molecule of nitrogen, and hydrogen is produced as a byproduct during this process as we see in the figure, but it is catalyzed into protons and electrons back by an enzyme called as hydrogenase. Apart from this nitrogenase, some of the microbes are characterized by the presence of an alternate nitrogenase. Sometimes it is called as unusual nitrogenase. Instead of molybdenum, vanadium is present in this unusual nitrogenase. For example, microbes like Acetobacter vinylandi, it depends upon this vanadium nitrogenase for fixing atmospheric nitrogen under conditions of molybdenum starvation. Apart from reducing nitrogen to ammonia, the nitrogenase can also able to reduce acetylene to ethylene. Henceforth, this reduction process is used as an assay for nitrogenase activity, which is called as acetylene reductase assay for estimating the nitrogenase activity. Here, the amount of acetylene reduced to ethylene can be estimated in a gas chromatograph. So, now how nitrogen fixation happens in root nodules when we talk about legume rhizobium symbiosis. So, the glucose 6-phosphate, which is the photosynthate obtained during the process of photosynthesis, act as an electron donor. The sucrose that is synthesized in the leaves and roots will be converted into glucose and fructose, which is then converted into glucose 6-phosphate by the glycolysis pathway, and again this glucose 6-phosphate is converted into phosphogluconic acid, which releases a molecule of NADPH. This NADPH donates electrons through to ferredoxin, which is a cofactor in the electron transport chain, and the ferredoxin is reduced. The reduced ferredoxin acts as an electron carrier and donate the electron to the iron protein complex of the nitrogenase enzyme to reduce it. Thus, the electron released from the ferredoxin is thus oxidized. And this picture, it shows a clear cut process, nitrogen fixing process that is happening inside the root nodule. So, the photosynthase that is assimilated as carbon substrate, they are the sources for contributing these ATP molecules or energy molecules, and the carbon dioxide is fixed by this carboxylation reaction, and during this carbon substrate utilization, electrons will be released, and this electron will be used to reduce the iron protein complex of the nitrogenase, which in turn reduce the nitrogen to ammonia. Here we can find a component called as lead hemoglobin, which scavenges the oxygen from entering inside the nodules. Why this mechanism functions here? Because the nitrogenase enzyme is highly sensitive to oxygen. To protect the sensitive nitrogenase, the root nodule tissues and lead hemoglobin are there to protect it. So, by this an anaerobic environment is maintained in the nodule through the collection of free oxygen by a heme protein called as lead hemoglobin. In addition, oxygen does not diffuse into the nodules easily. A thick layer of cells act as a barrier to oxygen diffusion. So, overall reduction process is here the dinitrogen is reduced to diamide, then to hydrazine, then to hydroxylamide, then finally to ammonia. So, as a conclusion, Biological nitrogen fixation accounts for 70% of N fixation by diisotrope. Rhizobium is free symbiotic nitrogen fixer. Apart from that, we are having free living nitrogen fixer, associative symbiosis as well as endophytic N fixers. And legume rhizobium symbiosis is a well studied and established one for nitrogen fixation studies. Nitrogen is an enzyme responsible for nitrogen fixation, which is having two units, iron protein and iron molybdenum protein, which in turn have 4 subunits. This nitrogenase protein enzyme is encoded by genes HDK, NIF HDK, and nitrogenase is sensitive to oxygen, and leg hemoglobin protects the nitrogenase. 6 ATP molecules are required for fixing nitrogen, and N fixers can replace chemical nitrogen requirement by 20 to 40% and increase the crop yield. Sir, முடிஞ்சது.